Hey everyone, just going to record a quick lesson here for you on the beginnings of cellular respiration. In particular, we're going to start to talk about really quickly the structure of the mitochondrion, where most of the process of cellular respiration takes place, and then we'll get into talking about aerobic cellular respiration, which takes place in the presence of oxygen. I'm not going to spend too much time with you looking at the mitochondrial structure. I'll let you have the opportunity to read through this quickly. Just be aware that there's lots of mitochondria inside cells, and there's sort of main structures that we're really going to pay attention to. The mitochondrion has an outer membrane, which is labeled by number 2 here. It has an inner membrane, which is sort of this wavy kind of pattern that goes all the way around here. It has, and this will be the site where the electron transport chain and chemiosmosis takes place. It's labeled by number 1 on here, and you'll see that when we get a little bit further on. There's what's called an intermembrane space, so that is the space between the two membranes, and it's sort of colored in this gray-white color that you see here. And this is where a concentration gradient is built up prior to chemiosmosis, which we discussed in photosynthesis. And then secondly, there's a mitochondrial matrix, and that's this orangey color in the middle here. And that's where pyruvate oxidation and the Krebs cycle take place, which are both parts of the process that we're interested in here. Okay, so there's two types of cellular respiration that we need to be aware of. One is called anaerobic and one is called aerobic. We're going to really focus in on aerobic right now, and later on we will get into talking about anaerobic. That'll come up in a concept later on. Aerobic takes place when there's ample oxygen around, or anaerobic will take place when there's very little, if any, oxygen around. So we're going to focus in on aerobic cellular respiration. We're going to break it apart into 4 stages, and today we're really just going to focus on these 3 stages: glycolysis, pyruvate oxidation, and the Krebs cycle. And then tomorrow, when I'm back and we're through what we need to get done, then we'll focus in on stage 4, which is the electron transport chain and chemiosmosis. What you need to remember is that cellular respiration is a process that uses glucose and oxygen and breaks it down into carbon dioxide, water, and energy. And as we've talked about so far, energy in this case is in the form of ATP molecules, which we know to be our cellular fuel. So anaerobic cellular respiration, I'm going to skip over more or less for now. We'll just take a quick moment to say that it takes place in the absence or very limited amounts of oxygen, and only has 2 stages as opposed to the 4 that we saw up here for aerobic cellular respiration. And there's 2 kinds that we're going to focus in on, and again we'll talk more about these later on when we get into the last concept in this unit. There's one that's called fermentation, and another that's called lactic acid fermentation, and then sort of alcohol fermentation. So one is going to produce ethanol and one is going to produce lactic acid. And like I said, we'll build up to that and talk more about it later on. So I'm going to basically spend the remainder of this time just focusing in on the first 3 stages of cellular respiration. Remember, I told you that this process is a process that gradually breaks down the glucose molecule. Rather than doing it all at once and possibly risking too much energy being released at once and not being able to capture that energy, the process of breaking the glucose molecule down happens in stages. And each time we basically make the glucose molecule a little bit smaller and a little bit smaller and a little bit smaller. And each time that we break a bond, a tiny bit of energy gets released. When that tiny bit of energy gets released, we try to capture it in some way. So we either capture it in the form of an ATP molecule directly to be used as cellular fuel, or we capture it in the form of these intermediates like NADH and FADH. So as we gradually break down that molecule, we're capturing those energy for later on to use. So we're going to start here with glycolysis. So in this diagram, what we're looking at is basically this is a giant cell around here, and I don't know if you can see my marker or not. And this is a giant mitochondrion, not nearly the size it would be in real life, or even in proportion to what a cell would look like. And then this area out here is the cytoplasm. And then we've got the parts inside the mitochondrion, again, greatly exaggerated so that we can illustrate what's going on. So we're going to start with the first process called glycolysis. And glycolysis is basically, there's that lysis again. So in this case, we're taking glucose and we're breaking it apart. In the process of breaking the glucose molecule apart, we actually need a little bit of help to start that process. And so there's actually going to be an input of some ATP into the process to give us the little bit of a kick that we need to get that started. And then the ATP gets used to break the molecule down. As we break the molecule down, we actually use 2 ATP to start this process, but then after it's done, we actually create 4. So there's a net positive of 2 ATP created. What results from this process is we get glucose being broken down into, I'm going to change colors just to make it a little easier for you guys to see. What we end up producing is 2 pyruvate molecules. And some of that energy that's created from breaking the bonds of glucose is also going to be used to create 2 NADH molecules. Now, I'm going to back it up for a second here. We know that glucose is a compound that contains 6 carbons. 6 carbons, 12 oxygens, 12 hydrogens and 6 oxygens. Pyruvate, interestingly enough, when we create 2 of them, they are actually 2 3-carbon molecules when we're done. So that process of basically splitting glucose into 2 3-carbon molecules, or splitting it in half, uses 2 ATP, but creates 4 ATP and 2 NADH. This ATP that we've created can go and be used elsewhere in the cell. The NADH, you'll see that the arrow over here points to the electron transport chain in chemiosmosis, which is the 4th stage of cellular respiration, so we'll catch up with that one later on. Those 2 pyruvate molecules will then move out of the cytoplasm of the cell and into the mitochondrial matrix inside the mitochondrion itself. So remember, the mitochondrial matrix is the central part, the part that is inside of the inner membrane. And those 2 pyruvate molecules will then go through a process called pyruvate oxidation. Okay, and it's going to create some interesting things here. First thing it's going to do is create 2 what are called, I'm going to have a terrible time writing this, acetyl coenzyme A, or we'll just call it acetyl-CoA. And each of these acetyl-CoAs is 2 carbons in size. So we went from a 3-carbon, 2 3-carbon pyruvates down to 2 2-carbon acetyl-CoAs. And the other thing then that comes out of this process is we're going to release 2 carbon dioxides as waste products, which we know to be waste products of cellular respiration. And that totally doesn't look like a CO2, but that's what it is. That's an O and that's a C. Maybe I can erase it if you bear with me, and I'll try again to make it look a little nicer for you. So 2 acetyl coenzyme A's are created, and then we also create 2 carbon dioxides from the process. So what we're going to see as we move forward here is we're going to continue to gradually break down that glucose molecule into smaller and smaller pieces, reducing the number of carbons in it. And each time we reduce the number of carbons, we create carbon dioxide, which gets sent off as waste later on. Also in this process, we create 2-NADH, just like we did up here in glycolysis. And you can see by the arrows here that the 2-NADH end up getting sent off to the electron transport chain in stage 4. So they're ending up getting sent off over here. Okay. Now, moving on to the third step. The third step takes these 2 acetyl coenzyme A's. Okay, so these two carbon molecules, and please don't get hung up on the name here. Just know it as acetyl coenzyme A. When you get into later biology courses, you can dig deeper into what this actually means and what it looks like. But for today, just know that there are two carbon molecules that feed themselves into stage 3, which is called the Krebs cycle. Okay, named after somebody by the name of Krebs. What happens in this process is each molecule goes through a cycle. It goes through a series of simple chemical reactions called oxidative phosphorylation. And what's sort of spun off of this cycle is 2-NADH, or actually 6-NADH, right? So 2, 4, 6, and 2-FADH, and 2-ATP. It used to be that you had to know the different steps involved in the Krebs cycle, and there were different pieces of this process. Right now, all we need to understand is that two carbon acetyl coenzyme A's feed into the cycle. It does its work and spins off 2, 4, 6 NADH, 2, 4, 6 NADH, 2 FADH, 2 ATP, and then the very last thing, again, as a waste product, is we eject from the process of breaking down the acetyl coenzyme A's, we eject 4 carbon dioxide molecules. Now those 4 carbon dioxide molecules join with the other 2 carbon dioxide molecules. That gives us our 6 carbon dioxide, which is the waste or one of the products of cellular respiration, and these exit the cell and leave the cell to enter into the atmosphere or wherever it goes. They enter into our bloodstream if you're a human or an organism that has blood, and then they get carried to the lungs, and then they get expelled in the lungs when we exchange them for oxygen. So I'm going to take a deep breath here and just run through this really quickly one more time for you. Pay attention to what's going on, and then what we'll do is tomorrow I'll run through this similar to what we did with photosynthesis. We'll run through it again tomorrow, and I will show you this whole process again, and hopefully it'll make a little more sense the second time through, and then maybe even a third time through later on. So starting in the cytoplasm of the cell, we have a process that's called glycolysis, and there's that lysis part again. Lysis means to break apart. So we are going to lyse or break apart the glucose molecule. Glucose molecule is 6 carbons in size. We're going to kick off the whole reaction with 2 ATP. Those 2 ATP molecules are going to power the initial breaking down of the glucose molecule. And that glucose molecule will be cut in half into 2 pyruvate molecules. Each pyruvate is a 3 carbon molecule. So we've taken a big 6 carbon molecule and broken it down into 2 smaller 3 carbon molecules. In the process, the glycolysis generates 4 ATP and 2 NADH. Okay, the ATP we said can be used again in the cell for various cell purposes. The NADH we said we will follow along with that molecule and see where it heads off once we get to stage 4, the electron transport chain. The 2 pyruvate molecules find their way then into the mitochondrial matrix of the mitochondrion, and those two pyruvate molecules undergo what is called pyruvate oxidation. My mouse has just quit on me. There we go. And pyruvate oxidation is the process of taking those two pyruvate molecules and breaking them down into two acetyl coenzyme A's, which are a 2-carbon molecule. In the process of doing this, two NADH are created, and we eject 2 carbon dioxide. Now, the two acetyl coenzyme A's then get fed into what's called the Krebs cycle. In the Krebs cycle, the molecules move around through a series of reactions, and we generate 2, 4, 6 more NADH, 2 FADH, 2 ATP, and we eject 4 carbon dioxide as waste from the process. Those six carbon dioxide molecules, the 6 carbons that initially started in the glucose molecule, are ejected out of the cell and released as a waste product of the process. When the dust is settled, we have created a net of 4 ATP. So we've used 2, but we've created 6. We have created 2, 4, 6, 8, 10 NADH and 2 FADH. Just like in cellular respiration, or in photosynthesis, these NADH and FADH molecules are short-term energy storage molecules. So they hold on to the energy that is generated as we break the bonds of glucose down. And so that energy is being held inside these molecules and will be fed into the electron transport chain in chemiosmosis to eventually be generated into ATP. We'll glance at that in the next lesson. For today, I want you to understand that the whole process of cellular respiration is the process of trying to take that original glucose molecule and a series of stages break it down, capturing energy in molecules along the way and storing it either for direct use in the cells in the form of ATP or to be fed into the electron transport chain to generate more ATP. So I hope that's okay for a little crash course in what's going on with cellular respiration. We'll again pick this up tomorrow. We'll run through it one more time. We'll go through the massive amounts of text that are in the following pages, which I basically just talked our way through, and then we'll get to the 4th stage, which is chemiosmosis. Hope your day's been well, and we'll see you again tomorrow.